good part of our group studies post-translation regulation very particularly. And so within that, we're focusing particularly in kinase signaling and to some other extent on ubiquitin regulation. And I'll just make a quick advert to a project that is going on in the group that has already finished, just as an advertisement. And so within the trying to understand protein post-translation regulation, in particular kinase signaling, these are different aspects of the work that we've done and continue to work on, trying to understand how throughout evolution, phosphorylation sites and kinase signaling has evolved. That has led us to the question of, how many of these phosphorylation sites that have been discovered in the last 5 to 10 years, how many of these have actually any functions, and what are the functions that they are doing inside cells. And then how do these kinase signaling systems work, and how these changes in either infection or cancer. I'll just make an advertisement to a project that was published recently, also in collaboration with the Pride team at EBI. And so in this project, the question was how many phosphorylation sites there are in human to begin, based on our current knowledge of mass spectrometry data, and then which of these phosphorylation sites have any relevance, or which we think are most important and worth looking at. And I won't describe the project in much detail, so just that to know that together with the Pride team at EBI, we researched deposited mass spectrometry data to identify on the order of 120,000 reliable human phosphosites. And out of these 120,000 phosphosites, we think only some of these are likely to have strong functional meaning. And so in order to understand that, there's around 2,000 phosphosites that we know have functions. And we use a series of features such as the degree of conservation, how often they are regulated across conditions, or the statistics of the mass spec data, and so on, to try to rank the phosphosites according to functional relevance. And the searching of the mass spec data was done by the Pride team, Andrew there, and it took quite a lot of compute power, not a simple task, to be able to run the search using MassQuant on this number of raw files and this number of PSMs that were identified. And in the end of this, we have an order of 120,000 unique phosphosites. And the purposes of this, of course, is we have to control the false discovery error that you'd get if you want to run a search of all these spectra together. But if you want to do a search individually and then try to combine the data, then the error rate would increase dramatically. And so if you're used to looking at phosphorylation data that may be in the Phosphosite Plus database, that database now may have accumulated a significant amount of error. So we would suggest that this is a better way of trying to get a consolidated data set. Then, as I said, if you're interested in phosphorylation, we have a series of evidence that suggests which of these phosphosites would be more likely to be quite important for the cell. So the most obvious one being, if a site is extremely conserved throughout evolution, then it's a higher chance than that this particular phosphosite might be quite relevant, such as, for example, this case over here, where you see a high degree of conservation, for example. And so based off of this, so based off of this idea, we can, we can now rank all human phosphosites according to their importance for fitness. And so we have this functional score over here that for which we can now rank all human phosphosites. And the important thing is we can, from the background distribution of all phosphosites, we can identify the ones that have a highest scoring score. And if you look at phosphosites that have a known function, or phosphosites that are associated with diseases, these tend to be more highly ranked by this approach. And so this is just a quick advertisement for this project. I'll skip to the other projects but in a sense, what we have here that I think is useful for many members of this community is that we have reprocessed a large amount of MS data to generate a comprehensive list of human phosphosites. And we integrate the different sets of features into a single score that can rank phosphorylation according to functional importance. This is based off of the idea that not all phosphosites are actually likely to have any function whatsoever. And we have other evidences from evolution and mutational studies to indicate that there are phosphosites that are likely to be not relevant, not doing anything. And so we can then start from the top of this list that we generated to identify important fossil sites. And so I'll skip now to the analysis that we've been working on relating to SARS, which I think this is why most people are here. And again, the caveat here being that this is a big project involving many people that was done in a very short amount of time. And so I'll talk also about the way, how interesting it was to be working in this mode. And there's over 100 people involved in this manuscript. And so there is the part of generating the network. There is now continuations on different fronts. So I am helping to organize the team that is doing the bioinformatics or systems level analysis view of this data and additional data sets that are coming. But there are other groups that are working on doing structural biology on the interaction between the viral proteins and the host. There are drug screenings and chemical biology analysis being done to try to identify new drugs to screen, and infection experiments that have been already conducted and are still being conducted. And this will in principle feed into clinical trials at UCSF and in other places. And so my contribution is confined to this, trying to organize different bioinformatic analysis that have been done on this first interaction map, but then also other data sets that are being generated by the different members of these teams. And so this is a large effort that's been done primarily by groups at UCSF in California, which where I did my postdoc, and this is why the connection. QBI, which is the Quantitative Biology Institute, is directed by Nevin Krogan, and this first study, the interactome study, is headed by that lab. And there have been two other international groups besides ours here at TBI and Cambridge, one in the Institut Pasteur, and the other at Mount Sinai, who have been involved in the drug screening effort. So I am not an expert in any way, neither in viruses, infection, or this particular coronavirus. Some points of note: this is a RNA virus, and it's quite big compared to other RNA viruses. It has many proteins. And I guess on one hand, this is also interesting because it means a complex way that this virus interacts and hijacks the cellular machinery, which may emphasize why it's interesting to target human proteins. Of course, you could also imagine targeting specifically the viral proteins, and that could be, on one hand, even more specific because these viral proteins do not share homology with human proteins, and therefore drugs that target these will be, more likely to avoid side effects. There is the idea, though, that if you target the viral proteins, you may end up having an easier route for the virus to escape because mutations in the viral protein that can escape the drug will be easier for the virus than if you're targeting the human proteins. Then it is unlikely that the human population will have variation that will make that drug no longer work. Of course, targeting human proteins has the side effect that it will cause potential issues. Of course, those human proteins have many other functions besides the functions that the virus uses them for. So there's a complex life cycle of replication of the virus inside the cell. It takes control of translation. It creates interesting replication areas for itself inside the human cells. And it also, of course, then needs to escape. It also needs to try to avoid the normal antiviral defenses that the cell has. And all these proteins that the virus have are doing many parts of this. So it was a big effort, primarily by the Nevin Krogan's lab, to try to obtain the interaction network for these viral proteins. 26 proteins were tagged, strep tagged, and this was done. They were expressed one at a time in hex cells. So this will be a kidney immortalized cell line. This is almost all of the viral proteins, not all. And then there was affinity purification in 3 biological replicates. And then this was then analyzed by mass spectrometry, and each of these was scored by a score that rewards reproducibility and specificity of the interactions. The reproducibility of the pulled down data is high for in particular, if you think of it from that is the pull down and not a quantitative measurement of protein abundance. and the replicates are on the order of 0.45 to 0.75 per pull down. And the end result of this is, we think, a high confidence in the sense that it's reproducible and stringent 332 human interactors for these 26 viral proteins. As I said, my job was more to help organize several of the analyses that were done in terms of the system level view, and I'll mention some results as well that are not in the paper, but may be interesting for other people to think about. So I won't talk about the standard gene ontology analysis, just gives you an overview of what are these interactors and what are the cell biological functions that for these interactors relate to. I'll talk a bit about the PFAM enrichments, the expression of these interactors in the human tissues. I'll show you some relationship with response to infection, and I'll also, towards the end, I'll show you some additional analysis that are ongoing and trying to compare this network with other networks from the viruses. So one of the things that is maybe something that you won't often do with a pull down, but it could be interesting to think about, is if you take a pull down of a given protein, you'll find many interactive partners for that protein. And then you can ask, are there protein domains that are commonly shared for the interactive partners of that protein? So on this graph, you're showing you on the y-axis, you have the viral proteins, and on the x-axis, you have the domains that are enriched. Sorry, the other way around. The x-axis, you have viral proteins. On the y-axis, you have here the protein domains that are enriched in the targets of that protein. And so what this shows you, for example, is NSP7 has a specific enrichment for having the interactors of the NSP7 have oftentimes a RAS domain, or the interactors of E oftentimes have a BET domain. What this suggests is that potentially, because NSP7 interacts with proteins that often have a RAS domain, that particular domain is the interface between the proteins that contain RAS domains and NSP7. So that allows you then to suggest a mechanism for the interaction model. And in particular for the E and the BET domain, there is additional evidence to suggest that this in fact is potentially the mode of interaction. I'll keep showing the names of people who have done this particular analysis throughout the paper. So Tina, for example, did this enrichment analysis. So one of the concerns that, of course, many people will have is that this interaction network comes from HEK cells, which are not the tissue of infection that this virus affects. And so one of the things that we did was to ask, are the interaction partners of these viral proteins likely to be highly abundant in different tissues? And so this, from the protein level, you can rank the degree of highly abundance of each of these prey genes. And here we're just scoring the deviation from the median of the interaction partners versus the background distribution of all proteins. And just ranking this, you can see that the interaction partners of the viral protein actually tend to be more abundant in the lung than in other tissues. And then it goes down the list to other tissues. I don't mean to say necessarily that this is the absolute ordering of this, but it just says that the lung tends to be a tissue where these proteins are highly abundant. On the graphic to the right, you have the GTAX expression. So on the y-axis, the x-axis, you have just the degree of expression of each of these genes. So each dot here is one of the prey genes. And on the y-axis, you have the fold enrichment compared to the median of all other tissues. So if it's higher up on this graph, it means that it tends to be more specific to the lung compared to other tissues. If it's more to the right, it just means that it's more abundant protein in the lung itself. So anything that's above 0 here tends to be more specific to the lung. And each dot here is an interacting protein of the virus. And in red, we're coloring here the ones that are the drug targets, so potential drug targets, for which there is a drug that one could use to target this. And what's the point of this is to show that many of these tend to be specific or more specific to the lung than other genes, which you think is an interesting point to say that on one hand, these viral proteins are interacting with things that are likely to be expressed in the lung, which is the relevant tissue. And on the other hand, if we were to drug these genes, then it's likely that this will be a safe target. because these are likely to be less expressed in other tissues, therefore maybe less important. One thing that we looked at, this work done by Maria and Gianluca, is that we also looked at differences in expression across gender and age. And I think many of you will be now know that this is a disease that affects males more strongly than females. And there is still no idea why. Of course, this could be, many issues have been described. In China, men tend to smoke more. So that was one of the first hypotheses. But this is no longer seems to be a valid hypothesis, at least in other countries. The same ratio is observed, and there's not such a striking difference between male and female in terms of smoking. And so one possibility would be that some of these interaction proteins are differentially expressed in the lung in males and females, in particular older males. And there are 12 of these genes that are differentially expressed towards older males versus older females, and that could be an explanation, but we didn't really follow through on this yet. So, but that's something definitely that's has not been figured out, and I think it'll be interesting in the future if somebody comes up with a good hypothesis of how this could be. And so the other question that we looked at is there was this recent study looking at the protein abundance differences along the infection that was talked about 2 weeks ago, the last seminar. And so we compare the interactions that were derived here in this study with the differences in protein abundances along infection. And there isn't a significant difference, just 80 of the human proteins are regulated upon infection. There's a slight enrichment, is not significant, but what we've done instead was to compare changes in protein abundance of the human proteins and the corresponding viral interactives or the precursors that were measured in the same study. The idea was if there's any significant co-regulation, so if the viral protein is going up in abundance, is the target interacting human protein also going up in abundance across the 4 time points? And so in here, we do see a difference, which is shown in a plot. So the prey interacting proteins tend to be more co-regulated during the time course than average pairs of proteins. So we think both this and the tissue data, for me, gives you the suggestion that although this is done in hex cells, this is relevant for the context of the tissue, both the tissue and the context of infection. So I think that we do have data for many other pathogens, so physically protein interaction data for other pathogens. And out of those interacting proteins, 177 interact with one other pathogen, and 33 interact with at least 3 other pathogens. And so these 33 are, of course, very interesting because they may be either restriction factors or things that facilitate the virus infection in some way for many pathogens. So therefore, these are more likely to be functionally exciting. Another analysis that was done was just look at the evolution of the proteins that are interacting with the virus. So one observation, but not a very strong observation, is that these interacting proteins tend to have less missense variance in the population. But it is true that this could be biased by protein abundance. So the fact that this is proteins that come in a pull down, they will typically have a higher abundance and be more structured than the average proteome. There was an analysis that was not described in the paper, but it will be in the revised version, is that Janet Young had looked for signs of positive selection. So that means actually the opposite of this, is there signs that the proteins throughout recent evolution in primates have had a fast change in their sequence. This fast change could be due to many things, but one of the things that it can be due to is trying to avoid interactions with pathogens. Of course, this particular coronavirus is not a human virus. It's just only recently became a human virus. But there will be other pathogens that could have shaped this positive selection. And if this particular coronavirus is interacting with these proteins that are in the positive selection, then this may tell us about, again, these 40 particular ones may be more interesting because they are being selected to diverge quickly throughout recent evolution. And so the end result of this effort was this map of virus-host interactions. We think it is relevant for the infection, despite being done in hex cells. because of the tissue expression patterns that I've described, and also the fact that these proteins tend to be co-regulated with the viral proteins during the infection time course. Really the objective of this study was to try to find drugs that are FDA approved such that they could be as fast as possible translated into a clinical trial if we find something that is relevant. So the point was, once you find one of these viral interaction partners, if there is a drug that is FDA approved or close to being a clinical trial drug, then this could be tested in the lab for, first of all, for an effect on cell lines, but then from then on, it could be translated into potentially clinical trial. This was done through a lot of curation effort primarily. And I'll just show you one example, but there are many such examples described in the paper. So here's the N protein that interacts both with stress granules, but also with RNA processing proteins. And this includes the kinase 2. And the idea here could be that these directions are in some way related to stress granule formation, and then that would have an impact on this RNA processing machinery. So maybe casein kinase 2 is involved in this regulation, and therefore if you inhibit casein kinase 2, you may have an impact on the viral replication. It is important to note that I think many of these associations, although are very likely to have a functional impact, you don't know a priori if you inhibit this, is it going to actually increase replication or is it going to decrease replication? So depending on the fact that maybe the interactions here with the virus are meant to shut down the protein or meant to use its activity. So depending on those two factors, the drugs may actually exacerbate the infection. Okay, so based off of this, there was a list of many drugs that have been proposed in the paper. So this list has been revised slightly as well. Many of these are already approved or have been in clinical trials or preclinical trials. So in principle, if some of this is useful, then some of these could move quickly, which is the main objective of the study. So there's been, it's not in the paper, but there has been 40 of these have been now studied in cell lines. There's a number of these that have effects. Some are positive, some are negative, some are potentially exciting, I think promising. And just stay tuned because the data will be available in a revised version of the manuscript, I think, quite soon. Okay, so another point that I thought it was quite interesting to note is that this is a very interesting way of doing science. You know, there's over 100 authors in this author list. David Gordon is the first name on this list, but there's Gwen, Maddie, JV, and Kirsten, Chris, and Matthew are here as lead authors on the paper. And of course, many small and big contributions have gone into this, split into sub-teams. And it was done in an incredibly short amount of time. So I think it also shows that, you know, when people are focused and interested in trying to do at least their bit of contribution, you can really do useful science in a short amount of time. I've been told that the constructs for expression of the viral proteins have been sent to over 100 different labs so far. So, and I think the preprint has already 6 citations. So I think I'm pretty sure that this is going to be reused by a lot of people. Okay, so in the next just a few slides, I'm going to give you an update of something that we are working on just to expand on this. And it's a fairly obvious thing to do, and I think other people will do it. So as you may have noted, as I described, the interaction network so far is focused really on just the direct, strong physical interactions between the viral protein and the human proteins. But there's no reason why we cannot try to expand this to other protein-protein interaction that are second neighbors of this protein interaction. And those could be interesting because there may be a drug for this, not the first interactor, but something that's commonly known to physically associate with that. And the other thing that we didn't do quite enough of was to try to compare the interaction network of COVID-2 with other pathogens, and then to use this to study the cell biology of this particular virus, but also to propose additional small molecules that one could use. This was done by Inigo in our group. And so our strategy to do this was to try to expand on the network and to try to identify modules, so highly connected protein interaction network modules that are interacting with the SARS protein. And the way we've done this, there are other ways of thinking about the problem, but you just simply take the human interacting proteins that have been identified in the study, you place them in the interactome, and then you propagate the node weights to the nearby interacting proteins. So this is how it would look. You take this would be the first interacting partners of the virus protein, and then you propagate the weights in the network until you then define a group of proteins that are have shared interaction partners with the viral interacting proteins, which would be one of these modules highlighted with a circle. Then I think the methods, there are many methods to do this sort of approach, and I don't think they often don't change substantially too much what you end up back. So after this process, you end up having these modules, which one can call a pathway or a complex. This is not so different from doing a gene set enrichment analysis. The advantage being that we are not so biased by the gene sets that are in Go that have been curated by people, and of course, you will be biased by the interaction that you provide to this. And we've been using this in a different context, and so I think we have some experience on how to deal with these problems. So once we get these modules, then we further prune them to retain only really strong physical interaction networks. So we've done this for different pathogens. And then for each of the subnetworks, we can compare all possible subnetworks between, say, SARS and all the other pathogens. And then we annotate the subnetworks by doing gene ontology enrichment analysis. This is just to give you an indication of what types of genes are in there. And as I said, then we prune the edges that are only high confidence physical interactions. So there has to be at least one evidence coming from pull down, and it has to have at least 2 different experiments supporting that interaction. This is just an overview of the types of cell biology that we find commonly across different pathogens. So this is then, for each of these pathogens, you have a list of modules, and this is just annotations of what these modules contain. So for example, proteasome biogenesis and viral expression and translation initiation, RNA splicing and ubiquitin are often things that are often interacting with viral proteins. And further down, you have things that are more specific to one or other pathogens. TNF alpha signaling, again, is something that often is used to try to stop the viral antiviral response. So then we get a map of this, which I don't expect you to look at. I hope you can at least see some of the coloring. So you have here some of the viral proteins from SARS, from SARS-CoV-2. And then in yellow would be the interaction partners that were identified in this first study from Gordon et al. And the other edges are edges that expand to nearby protein interactions that we think are part of the same related functions that the same proteins are part of. And so the edge around the colors, the edge around the proteins indicates also that this particular protein is also pulled down with other pathogen interacting proteins. And so these groups of modules that I'm showing you here right now would be modules that are associated with COVID-2, but also other pathogens. And then these other 3 modules will be protein modules that are more specific to COVID-2. It doesn't mean that some of these proteins do not interact with other pathogens. It's just that there's no enrichment for this particular group of proteins to be interacting with other pathogens. And just a high-level overview of what these processes are is shown here. So, as I said, things like protein ubiquitination and control the kappa beta pathway, mitochondrial translation, RNA processing, translation, RNA and export from the nucleus, control of the cell cycle and respiratory chain will be things that are common to many pathogens. And then ERADs will be things that are a bit more specific to this particular virus. Also, spliceosomal assembly is or RAS signaling is part of something that's a bit more specific to this virus. So as a zoom into one of these submodules. So here we have, for example, interactors of WRF10, but also interactors of NSP15 and another protein. And what you get out from here is you can, if you know some of these genes, these are often things that relate to protein regulation and calling, and also NF-kappa beta signaling. And this was already highlighted in the previous paper, where ORF10 was interacting with several members of CoL2 complex, and through the expert knowledge, you could already indicate that there is an enzyme that is required for the function of CoL2, and there is a particular drug that one could use to inhibit this protein NAE. And so this was derived based off the expert knowledge of knowing this particular complex. In this case, by doing this network extension, we could also suggest already that this particular drug could be used, even though this is not a direct interaction partner of this complex, but is strongly associated with the complex, such that one could suggest this drug. And so there's another example here where one could suggest another drug that could be used, or another protein that has drugs that one could use to target the function of this group of proteins, and hence we think could also be used to then target the viral replication. Another interesting aspect that I only glossed over was that this virus and a few other viruses create replication zones by hijacking the redirect pathway. And this is, we think, an example of this group of proteins, this module that is interacting with M and NORF8. This is the group of proteins that is likely to be involved in this process of creating this double membrane vesicles that are required for this replication areas for the virus. And again, here we can propose at least one other protein that is not directly physically interacting with the virus, but has a series of potential drugs that one could use to try to impact on the replication. Now, just for the limitation of time, I'll just skip but essentially this is related to a biological process that I am not familiar with whatsoever, but where these vesicles that are related to ERAD functioning can be hijacked by the viruses. And this is, in this review, they talk not specifically about the coronavirus, but is a similar process whereby these viral proteins, this is not coronavirus, but it could hijack these vesicles for their own purpose. And so there has been one other study done on coronavirus 1, not coronavirus 2, where they've done bio-ID tagging to identify proteins that are involved in this microenvironment for replication. And these proteins circled in red are all found in this bio-ID study, which again further suggests that this network expansion is correctly identifying relevant proteins for the virus to work with. Okay, so in this second bit that I told you about, we have this human-viral interactome. We did this expansion, and we filtered down to, we think, is a high confidence set of proteins that are not the direct target of the virus, but then they are part of the same protein complexes, and therefore could be used also for drug treatments. And so this is what we're doing with this, and we'll try to, in fact, try to wrap this up quite soon. But other aspect that we're also working on is not just mapping to these genes the known drug targets, but also maybe using drug screening data and CRISPR data to try to target, make predictions about other molecules that could also target the same genes. And we're doing a bit of structural modeling for the human viral interactions. And that only gives you just a higher confidence to say if we have a putative interface that we can use, then this gives us highest confidence that this is a true interaction. Okay, and with that, I finish. I have here a series of acknowledgements. I'm happy to try to take questions if I can.
Okay, thank you ever so much, Pedro. That was really, really fascinating talk. Yes, for the questions, we will work with Slack. If everyone who is in there just wants to head over and ask any questions they've got at this point. Also, feel free to react to any questions so we can prioritize. We've got quite a few coming in. So I will start with a question from Andy Pitt. He has asked, where interactions are with larger known protein complexes, can you narrow down to the specific interactions? Where interactors are where interactors are in more than one process, this should be possible.
Not sure if I understand the question. Can we figure out which is the direct interaction partner and which are secondary interactions coming from the pull down? if that is the question. I think it's not often possible unless you can identify an interface that is a plausible interface. There are many difficulties with this. So unless you actually have a structure, and there are very few structures, there is no sequence homology. So if you simply try to do homology modeling of the interactions, you come up with basically zero, because the viral proteins are not homologous to proteins that for which you have a structure in complex with human proteins, I mean.
Okay. Is that roughly what you're asking, Andy? I don't know if you're still on there and you can just give us a nod or to add anything else. I think possibly what he's trying to say is can you rule out protein interactions being between direct interactions or possibly via larger indirect interactions because of perhaps sort of covalently linked proteins?
Yeah, so that is essentially what I was trying to say. So,
okay.
So the answer is no.
Okay. Okay. Brilliant. So we have a couple of questions from Harry. Will you generate the functional database for any other post-translational modifications? I assume this is referring to earlier parts of your talk, and for crosstalk between post-translational modifications.
So we have some work on ubiquitin, trying to rank ubiquitin sites. Is more complicated because ubiquitin is often linked to degradation, so that makes it a bit harder, but we have some work on that.
Okay. And Susan Akalo has asked, what do you think about the role of epigenetics in the host response?
Is this from the point of view of having a change in the cell during the infection?
She's followed up with saying there are a few proteins with direct interactions, BDR2, HDAC2, but some networks, stress granules, inflammatory responses have historically been modulated by methyltransferases, bromodomain-containing proteins, deacetylases, etc. Yeah,
so I have less of an understanding of how the epigenetic landscape itself of the genome and how the virus may change the epigenetic landscape of that cell. I have little knowledge of that. I mean, many of these domains and binding domains could also be, used for other purposes, not necessarily to change the epigenome itself. So, but no, I guess I have no idea. And as far as I know, there's yet no study out there on this.
Fair enough, fair enough.
There is studies on changes in gene expression programs. I think that is the most obvious thing to look at.
I'm just going to jump over. I think there's some questions over on the Zoom chat. We've been asked, is there any specific affinity for spike proteins, a virus specifically for lung cells? How do they interact with other cells?
You mean the ACE2 receptor expression patterns or other receptors?
I imagine that's what we're referring to.
Yeah, and so there has been a preprint showing, so in fact, the ACE2 is not strongly expressed in the lung tissue per se. It is overall not highly expressed. It's actually highly expressed in the heart, kidney, and I think the testes. And there is, however, a population of cells in the lung. There's a preprint, I don't know the reference by heart, but there's a preprint that had looked at this. And there is a subpopulation of cells in the lung that do have a high expression of ACE2, and that is thought then to be the target of this. And then I don't have direct knowledge of this, but there was also some discussion of the fact that inflammation may lead to overexpression of ACE2. This I am less sure about. There's also apparently some interesting expression of ACE2 in some particular cells in the nose, and that may have been linked to the fact that some people seem to lose their sense of smell.
Interesting, yeah. Okay. Got a question from Steve Harding. You talk a lot about protein interactions, but what techniques did you use to identify and quantify these interactions?
So this was APMS, so standard pull down followed by mass spectrometry. So we have the standard analysis where you take the fragmented peptides, you look for best match in the library, and you have intensities and replicates. And so once you have the replicates, you can do statistical analysis to make sure that, well, of course, on one hand, you have to do statistical analysis to make sure that the peptides that you find are not just false positives. That's standard MS analysis. And then you have statistics to make sure that the proteins that you're identifying are reproducibly bound to that viral protein, and then specifically bound to that viral protein and not just the background. And that, there's already been quite a lot of work done to make sure that these analyses are solid. So I didn't go through into detail, but I'm pretty confident that these interactions are reproducible and specific.
Yeah, with the number of proteins you see, I imagine you don't have that much non-specific binding happening. So we've got a couple more questions. Harry's asked, these experiments were conducted in single protein expression systems,
Yes.
i.e. one viral protein per cell. Does interactions between viral proteins make a difference in the host proteins they're complex with?
Yeah, I should have mentioned that. So I think that's a big caveat, and it's a very important caveat. So ideally, you'd have some way of doing interactions for context of infection with a full virus and everything. So, as example, NSP7 and SP8 form a complex. And so, quite likely the interaction that they have will depend on having the other proteins there or not. So, this is a big caveat. It was pointed out by the reviewers as well, that I think one answer to that is, of course, each of these proteins often will have its own functions without the interaction partners. And so this is just a stab at that.
Do you know if there's any follow-up work being done to, say, do the same sort of study with infected cells?
I think there are a couple of other interactions projects ongoing, including a bioID study and other yeast-2 hybrids and things like this. I don't know of an actual, because you would have to tag the proteins in the virus without disrupting the proper transcription translation of the viral proteins, and that is probably going to be quite hard to do.
So another question from Rob Ewing. Are there sets of protein interactions from lung epithelial cells that you can compare to the HEC interaction with those that occur in the lung alveoli?
No.
Okay.
But it is a fair point. I think, again, this is a starting point, and doing this in other cell types, in particular more relevant cell types, would be a good idea.
Yes, I think you get a pass given the rapid nature of how quickly the work was done. Okay, I'll just ask a couple more, if that's all right at your end. So how this study could be extended to know the effects of the suggested drugs on different human races?
So the study that was already extended. So there's about 40 drugs that have been tested for their capacity to change the viral replication in cell lines. This was done in Pasteur and Mount Sinai, and there were several drugs that had an impact on the replication, and this will be in the revised version of the manuscript. And so from there, of course, ideally, some of these will be quite safe and readily usable in patients.
Okay, and I think I'll just ask one more question. Is there any knowledge of the stoichiometries between host viral interactions, e.g. how many viral proteins are there per its human target, e.g. in the lung?
No, I think there's none. But I should also follow up from there. I did mention that there is actually quite a number of structural biology groups at UCSF that are following up on this. So they're gearing up to do protein expression, purification, EM and X-ray studies. So there will be a continuation of this, and this types of question will be something that will be part of those studies.
Fantastic. Well, thank you ever so much, Pedro. Lovely talk. Thanks for asking all those questions, and thanks everyone for joining us. Please head over to the Slack channel if you have any more questions, and we will forward those on to Pedro himself. And that's all for today. Thanks to everyone who's made this possible, and we'll hope to see you in a couple of weeks' time. Okay.
Thank you, everyone.
Cheers.
Thank you for coming.